Well, good morning, everybody. I remember last year I presented from the basement of my house in the United States, and I think my program started around midnight. So this is much better, much better hour and much better company this time. So as Roland said, I'll be presenting on, updating on you on some tomato work, and then focusing a bit on our, our progress on insect resistance. So this is the structure of the presentation. I'm going to briefly highlight some features about the APSA tomato lines, and then I'll move into the update on the insect resistance, discuss a little bit about our heat tolerance magic population, our progress on mapping virus tolerance, brief things about the pipeline, and that'll be it. So this year we're offering APSA 12 tomato lines, which can be grouped into 4 groups basically of sister lines. There's about 3 sister lines in each group. Three of the groups are fresh market types, and 1 group is dual purpose type. This group are coded CLN4270J or various codes. There's 3 lines of this type. These are mainly semi-determinate. And what I like about these lines is that one thing I like about the lines is that they produce very good, vigorous foliage and then excellent foliar health. And we've noticed in Taiwan that early blight tolerance of these lines is really good. So I like that. The fruit are kind of a blocky round shape, as you can see, 100 to, let's say, 130 grams in range. Really nice, smooth shoulders, excellent internal and external fruit color. That's due to presence of the crimson old gold gene. And they carry a package of TY and bacterial wilt resistance. So the second fresh market group are coded 4256 or 4273. These are determinate and mainly determinate lines, very vigorous growth, some heat tolerance. Fruit sizes range from 120 to about 140 grams. They also carry TY and bacterial wilt resistance. And the third group of fresh market lines really were designed to improve the bacterial wilt resistance. And we were thinking especially of West Africa, but these are very applicable to other parts of the world. They're coded 4398 or 4398 generally. So we're trying to achieve levels of bacterial wilt resistance equivalent to the best source, which is Hawaii 7996. So the fruit weights range up to 100 to 120 grams or so. Very good foliage, nice fruit shape. The main feature, probably the negative feature about these lines is they're a bit soft. So they may need some work to improve the firmness of these lines, and they may be better of more immediate use in a trait development program or perhaps rootstock development. And then the fourth group are dual purpose lines. They're coded 4279. They range in size from 100 to 170 grams, mainly semi-determinates or mainly determinates. They carry fortified bacterial wilt resistance. So they are homozygous for BWR12 and BWR6 and are oblong in shape. They have excellent firmness. They carry high pigment gene as well as the crimson old gold. So the internal color is very, very nice. So now I'll move into updating you on our tomato insect resistance. So that's been ongoing at the center since 2014, and initially that work was led by Mohammed Raqqa. And in the early days, what he did is screen our gene bank accessions or selected gene bank accessions and identified whitefly resistance. So starting first with whiteflies, and he identified several accessions of Solanum galapagense and 1 accession of Solanum habrocaides. And I think you realize that there's no resistance to insects or appreciable levels of insect resistance in cultivated tomato. So our interest was more along the lines of Solanum galapagense, which is very closely related to tomato, and that's a big advantage. Galapagos resistance to whiteflies and insects in general is conferred by acyl sugars produced by high densities of type 4 glandular trichomes. So glandular trichomes are either sparse or absent in cultivated tomato. So we initiated interspecific crosses to move that resistance into tomato. And a major QTL called WF-1, located on the bottom of chromosome 2, was identified here, and it had been previously published. So this is not only our group, but others have identified this major QTL. And it's associated with high trichome density, reduced numbers of eggs, and high whitefly adult mortality. So this is very, very important, probably a transcription factor. So by 2020, we had produced several cultivated tomato by Galapagos populations that had reached the BC3F3. So that's where we begin the story here. So we conducted some trials this year, the end of 2020 and the early part of 2021. And what we wanted to do is take the work into the field rather than continuing the work in greenhouses using no-choice assays and so on. We wanted to take these lines to the field, and with several objectives. One was to quantify acyl sugar production. So again, that's the major mechanism underlying resistance, and look at production at different plant ages. We're looking for lines that showed stable levels of acyl sugars. And also we wanted to evaluate their fruit and horticultural traits. So this, the latter part of 2020 to 2021, so October 2020 to January 2021, we conducted one trial. And we have a second trial that began in March 2021 that's ongoing. So we worked with 11 BC3F4 lines, and these lines are all homozygous for WF-1. In addition, we included the recurrent parent, CLN36A2C. And in the spring trial, we added in the Galapagos accession, which is the donor parent. So the trial was conducted with 3 reps in a randomized complete block design with 6 plants per rep. And total acyl sugar levels were measured on individual plants at several plant ages. So this is quite a bit of work for a small trial. So these are the results of the first trial. Again, this went from October 2020 to January 2021. And the vertical axis is the acyl sugar levels in micromoles per gram. And the bottom is the horizontal axis is the different sampling dates. And we did the first sampling date at 32 days after transplanting. The 0. Is that the, okay, yes. So here is they can't see that, all right. Okay, so the bottom line is the recurrent parent. So that's the check back, please. Okay, and you can see that the acyl sugar levels of the BC3F3 or BC3F4 were somewhere at that stage 5 to 8 times higher than that of the recurrent parent. And then by 48 days, it was around 3 to 5 times higher. And at that point, the levels dropped down at 78 and 94 days after transplanting, which might lead you to conclude that acyl sugar levels start declining as the plant gets older. But I want to make you aware of the fact that the temperatures started to decline around that same time. So as we go from 32 to 48, the average temperatures were 23, 22, then it dropped to 18 and then to 15. So very possibly there might be effect of plant age, or it might be also due to a temperature effect, or it could be both. So in this figure, what I wanted to do is just show you some of the variability. So these are means of selected BC3F4 lines. The bottom one is the recurrent parent. But just to show you that there was quite a bit of variability within lines, within most of them. So that offers opportunities to select, which we did. We made selections within lines, and hopefully that will work if there's still segregation for certain modifier genes within the within these lines, besides WF-1. So this is results of the ongoing trial, which was transplanted on the 10th of March. And this time, what we wanted to do, well, as I mentioned before, we added in the Galapagos accession, which is the origin of the acyl sugar trait, along with the recurrent parent, which is the other check. This time we did the sampling a bit earlier, so 13 days after transplanting, rather than waiting longer. Reason is because it's important that this acyl sugar trait be expressed early on, while shortly after transplanting, so that it helps repel whiteflies during the vegetative stages when the plant is more susceptible, I would say, to virus infection and development of very full-blown symptoms. So the Galapagos parent, as you can see, is quite high. It's about double that. This is the first day of measurement. So it's about 10, maybe 9 or 10 times higher than the recurrent parent. And most of the BC3F4 fall within the range of 6 to 12 micromoles per gram. And then in the second sampling date, you can see that the Galapagos jumps up quite high. It's already up to 42. The recurrent parent drops down to 1.21. And the Galapagos, the BC3F4 fall within the range of, let's say, 5 to 13, which it's still quite high. This is much higher levels than what you would find in a cultivated tomato. So I think the question now is whether these levels are really significantly high enough to repel whiteflies. So that's the next question that we need to answer. So we do plan to take other samples. The last samples were taken yesterday, 61 days after transplanting, and we'll do another time after that. So what do these BC3F4 plants look like? Well, this is pretty typical. You can see that they're indeterminate, semi-determinate plant habits. They're much bigger and much more vigorous than the Galapagos accession. They've picked up the TY and bacterial wilt resistance from the recurrent parent. But you can see that we have some challenges ahead in terms of low fruit set, overcoming low fruit set, and the small fruit weight, so that the average is maybe 30 to 35 grams. And I'll talk more about that later in the next presentation in the afternoon. So next steps, what we want to do is hopefully identify BC3F5 that are fixed for high levels of acyl sugar and insect resistance, and then cross these insect resistant lines as donors to create a BC3 or BC4F1, develop segregating populations, map insect resistance, especially the possibility of additional modifying genes, design markers, and then apply forward and background selection. So select for insect resistance, and apply markers to remove or to eliminate extraneous Galapagos introgressions. So after this spring trial, we'll start up again in the fall with trials of BC3F5 lines. In addition to monitoring acyl sugar levels, we want to really look more at insect parameters. So looking at whitefly eggs, nymphs, and also possibility of looking at Tuta, because now we have Tuta absoluta in Taiwan. Okay, now moving on to the magic population. Heat tolerance has always been a major objective of tomato breeding since the beginning of this center. And in 2017, we initiated the development of a magic population. Magic stands for multi-parent advanced generation intercross. And with two objectives in mind. One was to map traits underlying heat tolerance. So that's pollen viability, stigma exertion, flower number, anther dehiscence, and so on. And second was to identify lines with improved heat tolerance. So we started with 8 founder parents, 4 of them being heat tolerant lines. and 4 of them were disease-resistant lines. All of these are cultivated tomato. There's no wild tomato among this group. So the crosses were completed. We used a funnel scheme, as you can see on the right side there. We started with a base population, which we advanced by single seed descent. So at the moment, we're at the F5 and we're harvesting F6 seed. There is segregation in the population for TY resistance. And our plan now is to continue the characterization of plant and fruit traits during this optimal dry season, and then continue with phenotyping later on. So we've conducted whole genome sequencing of the parents just to make sure that we have sufficient SNPs to do mapping. This is work done by one of our postdoc, Yaping Lin, who's a bioinformatics specialist. So it does appear that we do have sufficient SNPs to do the mapping. Very interesting that the three of the heat-tolerant parents cluster together. Maybe no big surprise because they don't have any disease resistance to speak of, whereas the other parents cluster in different places. So this is, we've done some initial phenotyping. This is more under dry season. I wouldn't call these optimal conditions, but much better than what will happen in the summer months. And you can see that there's some interesting rills coming out of this population. We're seeing good distribution and good variability for a number of traits, for fruit weight, fruit set, flower number, locule number, and so on. So our plan is to phenotype these rills for a number of the heat traits during the 2021 summer in Taiwan. This is always a risky venture to do this in the open field in Taiwan, just because of the risk of typhoons. And but we're going to give it a try. We expect or hope to identify some of the better heat-tolerant magic rills that will multiply, and we'll be using some of those in the APSA heat tolerance project that'll be starting up in the summer. And then what we'd like to do is intercross some of the best magic rills, and along with other new heat tolerant lines that we've identified. There's some very nice F7 lines that I spotted the other day. And so intercross and then create a superheat population. You know, a few years ago, I think it was 2019, during the tomato diseases symposium in Taiwan, there was a trial of lines that were derived from a cross called CLN4197. And it was very evident that there was some of these lines were yellowing up and showing very strong crinivirus symptoms, where others were remaining green. So it made us wonder whether we might have stumbled on some tolerance to crinivirus, which would be special because I'm not aware of anyone who has really identified a crinivirus tolerance in cultivated tomato. But we didn't, at that time, we didn't know where that tolerance may have come from. So this summer or spring, we have screened the parents of this cross. And you can see symptoms are starting to develop on CLN on the right-hand parent there, CLN3447. So that's quite susceptible to crinivirus. And very clearly, the source of tolerance is 3764G, which is remaining quite green. These pictures were taken just a few days ago. So the symptoms are developing, but it does appear that there is something real going on in this population. So crinivirus it's an RNA virus. It's also vectored by whiteflies. And we see a lot of this now that we have the TY situation more under control. We see a lot more crinivirus in our trials. So you can see on the top, you can see photographs of what we saw a couple of years ago, which was the susceptible versus the tolerant. So our objective currently is to try to map crinivirus tolerance in this population. We're using a segregating F2 population and checks. And we've treated the F2 populations in 2 ways. One is exposing the F2s and checks to whitefly populations in the field under natural crinivirus pressure. And we've also taken a set of F2s and inoculated them in the greenhouse with crinivirus, virulifers whiteflies. And these plants will also be transplanted shortly. So it'll be interesting to compare these two populations. We're going to score them. We've devised a 1 through 3 scale, looking at spread of the crinivirus symptoms on the plant and also taking into consideration the severity of the symptoms. Our genotyping plan is to use, and mapping plan is to use QTL-seq. Okay, in terms of the next year and the years, a few years ahead, in fresh market tomato, the next group of lines that will be coming out will feature large fruit size and late blight resistance, pH 3 and pH 2, combining that with TY resistance and bacterial wilt resistances. So again, trying to really focus on some of these traits that the group has highlighted are very important. We have new batches of heat tolerant lines improved for fruit traits and multiple disease resistance, especially combining heat with TY resistance. That's really critical, but also with bacterial wilt. Combining tomato spotted wilt virus resistance with TY resistance. And then we have a few high rutin lines coming out for the dual purpose. So I think that is, okay, thank you. I'd like to acknowledge our donors and the group, my group that's worked really, really hard, working on so many activities. Okay.
Thank you very much. Our Q&A box has pretty much filled up. And I start, I hope, in the beginning with the questions. Are you comparing your acyl sugar? Are you comparing your acyl sugar measurements with visual ratings or with trichome density?
In the past, we also looked at trichome densities. In the field trials, we have not, but I would like to do that because I think it may not be just high acyl sugar content. It also may be a combination of high trichome density and high acyl sugar content. So that's something we want to add in the future trials.
How effective do you think these new whitefly resistant backcross lines in reducing leaf curl disease incidence under field conditions?
Okay. I would not depend on insect resistance or whitefly resistance as the sole component for TY. I think what we have to consider or think about doing is combining TY resistance, so resistance or tolerance to the virus, along with resistance to the vector. So the insect resistance, or at least the vector resistance, is a sort of a second line defense. And it will also help reduce the whitefly populations on the plants. And I think also will increase, and that will increase, I hope, the durability of the virus resistance.
Then there comes a question about the crinivirus. Which of the criniviruses did you detect? They use TOC, TIC. I think this is tomato chlorosis virus, yeah, and tomato
infectious. It's tomato. What we have here in Taiwan is tomato chlorosis virus. So that, of course, that prompts the question of whether this tolerance holds up against tomato infectious chlorosis virus.
And there's a question that relates, I think, to the presentation before, whether we are also working on diagnostic markers to detect the traits for introgression. Yes, in general, we are working, we are using marker assisted introgression. So why most of the WorldVeg lines are not introgressed with TY4, 5, or 6, and what is your experience on tolerance to leaf curl disease by TY5 and TY6?
Okay, last year we offered lines that carried TY3, TY2, and TY5. So we have lines that carry 3 of these TY genes. We're adding in TY6, so that's ongoing. We picked these particular ones because their mechanism of resistance differs, and I think their mechanisms complement each other. So especially TY5, which is a loss of function gene at the pelota, or loss of function allele at the pelota locus. I think that has a special mechanism of resistance that will improve overall tolerance. TY6 is useful. We're working on that now, as I mentioned before. We're at the F3, I think, evaluating F3 families.
Now a question to the WF1 QTL. Is this QTL dominant?
Yes, we don't know yet. We have not studied the gene action of this gene. That's something that needs to be done, of course.
Question on the target insects for your insect-resistant breeding. Which insects do you target?
Well, we started out with whiteflies, and we found that the resistance in Galapagos holds up against spider mites. It holds up against Tuda. I think the question now is, okay, the Galapagos holds up, but the level of acyl sugar may differ depending on which insect we're talking about. So we may have to establish thresholds or targets. So a certain level of acyl sugar may work against whiteflies, and what might work against Tuda might be higher or might be lower. So these are questions that we need to answer.
Lucy Sakai is asking about the seasonal and climatic effects on the acyl sugar production. How do these factors impact insect resistance?
Yeah, that's also a very good question. And that's something we're looking into now. So thinking about temperature effects. And well, of course, wet season will be a major factor, because under rains, we would expect the acyl sugar would wash off, or some of it. It regenerates, but still rainy season will be more variable. I expect there will be age differences. So depending on the age of the plant, I expect temperature to be a major factor. And also the position on the plant. So whether we're talking older foliage, younger foliage at the top, the levels of acyl sugars probably differ at different levels of the plant. So this is also something to look into.
And which levels of acyl sugar do you think are effective for insect resistance? From which level might be effective?
Well, that's something I mentioned before. We need to establish targets, but at the moment, our working hypothesis is 10 micromoles per gram. That's kind of what I'm looking for at least for whiteflies. And whether the caterpillar pests, whether that's sufficient, I don't know. If we can get a little higher than that, that would be good, 13, 15. But we don't want to make it so high that there's a metabolic cost to the plant. So you can have maybe we could develop a plant that is a super generator of acyl sugars, but there may be consequences on yield by having such high acyl sugars. So we need to kind of look at the consequences, the sort of a cost-benefit analysis of this trait.
Did you already compare now the acyl sugar levels in your lines with those lines that were produced by Martha Mushler?
No. We received some lines maybe 10 or 15 years ago, but we would need to grow those under the same conditions. But that also makes sense to include at least several of these. So Martha Mushler worked on different species. She worked on Solanum penalii, LA716, for quite a while, and that resistance is also quite good. It's quite effective, but it's genetically more complicated. And penalii produces different types of it produces acyl sucroses and glucoses. So it would be useful to include one of her lines in as a check, just for comparison. One of the problems we've had problems doing seed multiplication of some of the Pinalii lines so that they can produce fruit, but often there's seed production is an issue, at least here in Taiwan, even under the optimal season.
Excuse me, Dr. Roland, we cannot hear you when you ask questions, so you probably need to go closer to the microphone. Thank you.
Thank you for letting me know. Thank you. There is a question on how do you measure the impact of acyl sugar addition if your background is already strongly resistant to begomovirus?
Well, I guess the proof will be in the long term, whether it enhances the durability of resistance. But again, keep in mind that we're not just targeting whiteflies. We're interested in insects as vectors, but we're also interested eventually to looking at resistance to the caterpillar pests. So Tuta would be a big one, Helicoverpa, possibly Spodoptera. So it's maybe the immediate impact may be more there.
A question on the leakage drag. If you introduce higher acyl sugar, what's happening with the fruit size?
Well, what we've observed smaller fruit among these lines. I'm not convinced that is necessarily a linkage drag problem. It could be. And Roland has done some work looking at the association between whitefly 1 and fruit weight 2.2, and he didn't find any. They're not that. It's not a problem, right? So it could be a linkage problem, but it also could be just a lot of Galapagense. Even after 3 back crosses, there still may be a lot of Galapagense introgressions in these BC3. So that's something I'll mention a little bit later or in the next presentation.
Now let's change the topic. There are so many questions. We will not be able to answer all during the Q&A time. The questions that are left over, we will then address by email. How confident is it that the heat tolerance work well under Indian conditions? The heat tolerance you are working.
Well, that's part of the point of the project that we're going to be doing with APSA is looking at heat tolerance, expression of heat tolerance under different types of heat. So I think we all are aware that there's humid heat, there's very dry heat, heat that can go up to 40 degrees. Our Taiwan heat is more mid-30s, but very, very high humidity. So part of the objective of the project is to look at expression of heat tolerance under different environments and just see what happens.
And again, brief change of topic. Any update on early blight resistance in tomato?
Early blight is a disease. It's a foliar disease that we observe here in Taiwan, especially. Well, it's pretty much all year. We see it in both spring seasons and the fall season. It's something that I select against. So if there are lines that are severely or quite susceptible to early blight, I'll throw such lines out unless there's some redeeming trait about it that it might be useful in a breeding program. So it's a quantitative trait. There's a tendency for lateness associated with early blight. I think it would be useful in the future, and maybe the topic for another APSA project would be to screen our gene bank accessions, especially the Cerasiforme and the Pimpinellifolium, and look for tolerance to early blight. We have a very nice greenhouse assay for early blight, which it would be good, and we could screen a lot of accessions fairly quickly, and perhaps find some new sources of early blight, because I think most of us are depending a lot on the early blight resistance that was developed at North Carolina State, but we need to find new sources.
And any update on tomato brown rugose virus?
Okay, well, this virus has not come to Taiwan yet. I hope it doesn't, but we're preparing for it. I've heard that some alleles that the TM1 locus are effective. There was a report of resistance being a resistance factor on chromosome 11 that was mapped by an Israeli group. So we went back and looked for lines that carry TM1. We've regenerated, we're producing seeds of these. So we're trying to kind of prepare if this does come here, or if we do have opportunities somewhere in the region to select for it or work on it, that we have some materials to work on it.
Two questions on the magic population. First, was there any selection pressure applied during production of the magic population?
The only one that possibly there might be selection pressure for TY, because it's always present here, and if plants are infected early, they sometimes don't even produce any fruit. So I suspect we lost a few for TY, but we wanted to maintain a certain number and not just select. We could have selected out all of the susceptibles, but then we wouldn't know whether there were heat tolerant loci near the TY13 locus on chromosome 6. So we didn't want to do that. We wanted to maintain at least a population that we could possibly map such QTLs if they do happen to occur near chromosome 6 or on chromosome 6.
Second question to the magic population. The genome sequences of the magic population, suppose of the magic population parents available?
I don't know, I guess. It's something we haven't discussed, so we have to discuss that internally.
I think they're under production. We are planning to sequence those.
Well, Yaping has done it already, so at least, okay, anyway.
So they will be available soon.
Yeah.
Back to the insect resistance for advancement for back crossing. Did you use only acyl sugar as a selection criterion, or also directly insect resistance?
Okay, what Mohammed did, and Mohammed Rocco did most of the back crossing, what he did was generate a F2 or a BC1F2 population. And he would first examine trichome density for looking for glandular trichome density. He would pick out some of those that have the highest trichome density. Then he did a no-choice assay in the greenhouse, so reducing the number to a more manageable number. And then selecting the most resistance. So the no-choice assay, you actually look at different insect parameters, adult mortality, egg number, nymph number. Based on that, he picked 3 or 4 of the best plants and F2 plants. And what he did was he bulked the pollen from those resistant plants and used that bulk pollen to make the back cross.
But specifically from the PC3, from the PC4 to the PC5, did you also apply insect tests for selection?
We didn't, but we plan to do so. I mean, that's something we wanted to add in to the next trials that will start in fall 2021.
A question on virus complex that affected the tomato crop in Maharashtra during summer 2020. It should be a complex of CMV, GBNV, TMV, and tomato chlorose virus. Did you encounter this challenge anywhere else? This kind of virus disease?
So the answer
complex?
is no. I haven't. It sounds complicated.
Yes, many viruses at the same time. A question about the magic population. By what generation uniformity is achieved in a magic population, especially when you think of your tomato roots?
Well, we're going to start phenotyping in the F6. They may not be, there may be still quite a bit of heterozygosity. We don't know, but we can't, it would be nice to take them out to the F8 or even the F9, and maybe we'll do that, but it's a lot of work to advance these populations. And we need to get moving on doing, at least doing some of the phenotyping. So we're going to get started.
Now let's connect heat tolerance with insect resistance. Do you plan to combine insect tolerance with TY and heat tolerance?
Well, insect tolerance, we will combine with TY. We can't do field trials without TY tolerance with the insect resistance. But I think first we just have to focus on getting that heat tolerance or insect resistance into line. So the focus will be trait development. And then once that is settled, and once we get to development of lines that really start to look like a tomato, then I think we can think about combining that with heat tolerance. But at this point, we'll take it in stages, but there's certainly no reason why we couldn't use a heat tolerant parent as a recurrent parent when we make back crosses with the insect resistant lines.
Well, this is the point where I would like to thank the audience for so many questions. There are still 24 questions left over to be answered. And many thanks to Peter. Also, the audience is thanking Peter, says, Peter, very nice presentation. Thank you very much.